My name is Mixture Glory and my title for today's presentation is about Hematology Analyzer Sysmex XN1000. Whole blood count is one of the most common tests that ordered in the lab, in the clinics or hospital. It is used for follow-up for treatments monitoring, a routine screening or checkup. FBC test is run and ordered to evaluate based on the symptoms that shown by the patient. The analyzer that required running the test is called as Hematology Analyzer. Sysmex XN1000 is a benchtop hematology analyzer which is wide use in central clinics and hospitals, which is hematologist analyzer that has 6 parts and can perform up to 28 parameters such as RBC, WBC, platelet, HTC, HGB, MCV, MCH, and for the whole blood. It can be used for body fluid analysis which is up to 40 samples per hour, but body fluid analysis can be performed if the instrument installed BF mode. In addition, Sysmex XN1000 can perform another parameter like reticulocyte and fluorescent platelet if the system of RET and platelet F mode are installed like BF mode. Unlike whole blood, RET, platelet, and BF count quite cost. It means most of central clinics are not performing this test because of the cost of the reagent quite high. As for its principle and technology, it has 3 analysis principles to detect WBC, IG, NRBC, RET, PLT, RBC, HTC, RIF, and hemoglobin, which are the first one is hydrodynamic focusing method, also called as DC shed flow, is a method to detect RBC and platelet. RBC and platelet are counted by RBC detector via hydrodynamic focusing DC detection. The second method is fluorescent flow cytometry. It is used to detect WBC, IG, and RBC, reticulocyte, and IRF in the sample by using semiconductor laser. The scattered light that produced have its own function as shown below. As we look at the picture, forward scattered light is to provide the information volume of the cell. Lateral scattered light provide the internal structure information like the lobularity and granularity of the cell. Lateral fluorescent scattered light provide the RNA and DNA information that contain in the cell. Third is cyanide-free SLS method, which is also known as SLA hemoglobin method, is an adaptive method to provide hemoglobin analysis without cyanide reagent added. After RBC is sliced and the hemoglobin is liberated, SLS is added in the reaction chamber. Hydrophobic portion that reacts with the globin and the changes oxidize Fe2 into Fe3+ and binds with hydrophilic group of SLS. As a result, a stable reaction product forms. The binding of SLS and HGB concentration is measured using light absorbance at 555 nanometer. Hemoglobin analysis is calculated by comparison between the observance of a blank diluent sample with SLS HGB concentration. This is some of the safety information that we must be alert of and practice in the lab. As we can see, we have many hazards such as general hazard, biohazard, electrical hazard, corrosive hazard, and more. I will show you the marking on the instrument of Sysmex XN1000. Next, we go to the types of reagent. All the reagents have its own use, precautions, storage, stability, and disposal. The first reagent we have is cell-packed DCL, which is used to measure the numbers and size of the RBC and platelet. Next is sulfolyzer to determine the concentration of HBG in blood. Number 3 is lysocell WNR. It is used with fluorescent WNR to differentiate WBC non-basophil, basophil, and NRBC. Fourth, we have lysocell WDF. It is used with fluorescent WDF to differentiate count of WBC, percentage of neutrophil, lymphocyte, eosinophil, and monocyte. Fifth, we have fluorescent WNR, which is to stain the nucleated cells for WBC counting, basophil, and NRBC. Number 6, we have fluorescent WDF to stain leukocytes for determinations of 4 parts differential count. And last, we have Cell Clean Auto. It is a strong alkaline detergent to remove the residual reagent, protein, and so on from the system. Sysmex XN1000 is widely used in hospital and central clinics. It used to determine the patient disease based on the clinical symptoms that shown by the patient. Besides, Sysmex XN1000 also used in thalassemia screening, treatment monitoring, and annual screening, and so on. This machine can perform sample up to 100 per hour for whole blood sample. It is 6-part differential analyzer that can perform up to 28 standard parameters, while for body fluid, sample can perform up to 7 parameters, which can perform 40 samples per hour. And for your information, the application of the instrument is depending on the type of the sample. The Sysmex XN1000 is capable to perform analysis based on first, whole blood and diluted blood, and second is body fluid with BF mode installed only. General standard operating procedure. Firstly, check the instrument operation. Make sure the instrument is clean from any debris or any foreign object. Secondly, switch on the IPU. Thirdly, switch on the instrument by press on the on switch button located on the back sampler and in front of the analyzer just to open the top cover. Fourthly, perform quality control analysis and validate the QC results. Fifthly, perform the patient samples, validate and release the patient test results. Sixthly, perform any maintenance, which is daily, weekly, as needed. Number 7, perform any calibration if needed. And lastly, perform shutdown on the instrument. Next, we go to SOP, system cleaning. Check the status of the status indicator LED on the sampler and analyzer. Make sure the LED color is green color to start perform cleaning. Look the tube holder if not ejected out. Press mode switch to eject out the tube holder. And next, on the control menu, click the analyzer menu button and click maintenance. After that, click the cleaning submenu. Fill 4 ml of cell clean in the clean tube and place in the tube holder. Press start switch button to perform cleaning procedure. As for the SOP perform calibrator calibration, it is same. We have to check the status of the status indicator LED on the analyzer. Make sure the color is green to start perform the calibration. Look at the tube holder if not ejected out. Press mode switch to eject out the tube holder. Change the analysis measurement mode by enter the change measurement mode. For the calibrator calibration, select whole blood mode and then click OK. On the control menu, click the analyzer menu button. Click on the calibration and then click calibrator calibration. The analysis box will appear. Before place the vial calibrator calibration in the tube holder, mix it well and place in the sample tube holder. Press the start switch on the analyzer to start the analysis. Wait until the analysis is complete and the tube holder will slide out. This is important because we don't know when is the problem and troubleshooting might happen. When there is something wrong with the Sysmex XN1000, the alarm will beeping and help dialog box will come out. Based on the help dialog box, the error message list will display on the current error, while the action section will display the troubleshooting action that should be taken by the operator. Few errors that occurs is error related to pressure, error related to temperature, related to reagents and chambers, related to motors, related to blood aspiration, and more. In conclusion, hematology analyzes Sysmex XN1000 is very useful in lab, especially in disease diagnosis, treatment monitoring, pre-examination disease, annual screening, medical checkup, and so on. The Sysmex XN1000 is widely used in many countries because of the cost of the reagent and the throughput is worth. Sysmex XN1000 is capable to perform 100 samples per hour, is very suitable for clinics nor hospital that has high workload. To perform quality control analysis, maintenance, and calibration is also easy to perform. Sysmex XN1000 also offers another parameter test such as BF, RET, and PLTF mode, which are installed by the engineer based on the terms and conditions that have been agreed by both of sides. But the addition of other parameters is suitable for hospitals that perform many tests compared to clinics that only for primer.